How does spike protein contribute to the unusual clots? And also, what we're going to be learning in today's video, how does spike protein contribute to potentially dangerous inflammation as well and damage to our circulatory system? My name is Dr. Mikola Rashek of Neurogenomics, and we are investigating this series of information showing how spike protein might be contributing to the formation of unwanted and abnormal clots. We've already covered some of this information by one research group. Today we're going to be investigating other research group. They published some amazing information. These guys are from University of California, and both in San Francisco and San Diego. So let's get started. So clearly these authors already were aware of the fact that spike protein can contribute to abnormal clotting. However, they were mentioning that, hey, listen, this abnormal clotting is not just a problem with those who get acute COVID-19. Clearly other people can also have it, including younger people are also dying of issues such as stroke, clogged access of blood to the lungs, or heart attacks. And these are young people, even people who were asymptomatic with their COVID, or those who might have had mild disease. So this is something really serious. Obviously, then there's also the issue of long COVID afterwards. So not something we should take lightly or trivialize at all. So the very first thing that the authors did in this publication that I'm reviewing for you today, what they did is they wanted to see what happens when spike protein is being added to the blood of a normal healthy individual. So when they did that, they were able to observe that as a consequence of that, the spike protein increased the level of polymerization of the fibrin. So what does that mean? Basically, remember when I was reviewing what clotting does? It leads to fibrin molecules connecting with each other. When you have same molecules connecting to one another, that is what is referred to as polymerization. And when you add spike protein to the blood of a normal healthy donor, that polymerization of that fibrin is increased. So it clearly increases that level. On top of that, these people looked at these clots under microscope, and they use electron microscopy. So this allows us to to look at views that are basically as, nope, I'm not gonna be able to do anything with that hair. That allows us to look at views as close to atomic or molecular level as we currently possibly can. Oh my God. This is what happens when you spend a few days in the mountains. It's uncontrollable hair time. And what they were commenting is that these clots, the polymerized fibers, basically those wires or these spaghetti looking like noodles of fiber, they are thinner. They're also rougher at their surface. So they're not as smooth as normal fiber strings or spaghetti noodles in normal clots. They have other elements attached to them, and they're also are denser overall. So basically this is very similar to what these other authors were also observing as well. Okay, they also were able to mention that it is either the entire spike protein that can interact with fibrin, or S2 component of spike protein. So remember, spike protein is made up of two components, or two subunits, or two domains, I should say. So there's the S1 domain and S2 domain. So if you think of a spike protein where the top of it is like the mushroom cup, that's the S1 domain, and the bottom, the stalk part, the part that interacts with the membrane of the virus, or if it's expressed in our cells, interacts with the membrane of our cells, that would be the S2 component. That's the one, besides the whole entire spike protein, that is involved in the formation of these clots. is really neat. What they also did is they looked at the dissociation constant, basically how easily or how well the spike protein interacts with either fibrinogen or fibrin. Remember, fibrinogen is processed into fibrin. Fibrin is then used in the formation of the clots. And the dissociation constant for fibrinogen against spike and fibrinogen was 5 micromolar versus fibrin was 0.4 micromolar, I believe. What it means basically is the lower that number is, the better the interaction that is being observed between the two proteins. So that means spike protein interacts even better with fibrin. So these are very, very important, valuable data that these guys were producing. And before we move on to some of this amazing work that they did in mice, one more experiment they did on cells was that they were able to show that how much reactive oxygen species could be produced due to the exposure to spike protein. The reason why the authors wanted to investigate that specifically is because, and I didn't know this until I read this paper, is because fibrin itself also stimulates immune cells to produce reactive oxygen species. Now recall, reactive oxygen species are not welcome. They're not there. If we have them in too large quantity, they can be dangerous. So they might not be good. And what happened is when they expose spike protein to these immune cells called macrophages, those are the ones that often comes to the site of injury or a site of problem, such as a pathogen invasion. They're the ones who are can lead the charge in fighting with the problem on the site. They are the ones that can release those reactive oxygen species via one specific enzyme. We're going to be talking about that in a moment. And they were able to clearly show that the more spike protein that was available, the more reactive oxygen species is produced. Now remember, we talked about this. Reactive oxygen species are not good because they are so reactive, they can break a lot of things in our body, which is why we want antioxidants, because antioxidants will capture these. All right, so now let's move on to some of the amazing work they've done on mice. And they did a lot of great things. So before we go on there, a little bit of a background. They created pseudoviruses. So what does that mean? I reviewed that before in one of the videos. You can take a vector. So virus you already are familiar with. They use HIV, and the virus is of course manipulated in such a way so that it will not be able to replicate, but you can use it as a vehicle to introduce any protein of interest you want into it. And in this case, they were able to introduce the spike protein, right? And therefore this pseudovirus, so not a real virus because it doesn't behave like a normal virus anymore because it's been partially destroyed from being able to do that. And oh wow, sticky hair. My hair looks like fibrin spaghetti noodles. And they produced pseudoviruses expressing spike protein, and they introduced them to mice. And these mice, what they were able to show using fluorescent microscopy, they were showing that these mice were producing plaques, or basically they were producing, they had fibrin clot formation in their lungs. So they clearly were able to show that by staining with antibodies. So antibody had a fluorescent molecule attached to it, and that antibody can bind to whatever target you want, whatever the antibody is made to recognize and bind to. And this is how you can visually see in biological material where the protein of interest is being present. And they were able to show in the lungs of mice that fibrinogen was being collected inside the lungs. And in this case, they were able to show in like a red dye. Blue dye is usually, and we reviewed this in the past, blue dye usually will tell you where the nuclei of cells are. So that's where the genetic material is. This simply tells you hey, where are we finding this these proteins in a context of where the nuclei are. So this is just simply to be able to orient you. And then finally they were able to show that where that fibrinogen was being deposited also highly correlated with the presence of the spike protein. So in this case, the spike protein, they were targeting it with green fluorescent antibody, and fibrinogen was red fluorescent antibody. And when those two fluorescent molecules overlap, they produce yellow color. So that's how you get those images with a little bit of green, a little bit of blue, and then a little bit of yellow. So clearly spike protein is contributing this. Now, recall in a previous video I was mentioning that the authors believe that it's abnormal clots that are being deposited in some of the severe COVID cases we were observing early on in the pandemic. And so this helps to corroborate some of this information as well. But here comes the really cool next experiment they looked at. They took mice that are mutant. They were mutated not to be able to produce fibrinogen at all. So as a consequence, these mice cannot form clots. Now, what was so interesting about this, and I'm still learning about all of this, is that these mice, because of the fact that they don't have fibrin and they cannot form clots, also they don't have inflammatory diseases, and they do not have autoimmune diseases. So I thought that was super interesting, and we'll get back to that in just a moment. And sorry, I was running up a hill not long ago, and now I'm suffering consequences. And when these mice were exposed to that pseudovirus, that pseudo HIV virus containing spike protein, they no longer had the presence of inflammation. Oh, I actually should have mentioned before that, what they also, before they used the mice with the normal mice, so not the ones that do not produce fibrin, they also were able to show that macrophages also go to the same site where the clots are being formed, and they track those macrophages with a specific protein that is expressed on the surface of macrophages cells, of these immune cells. I believe it was called MAC2, MAC2. And this is also where they were able to show that presence of these macrophages also leads to production of reactive oxygen species at that very same site. And they tracked it through a specific enzyme that is responsible for producing that reactive oxygen species, which was nicotinamide adenine diphosphate oxidase. So that's a mouthful. The name is not that important, but what is important is that that enzyme produces the reactive oxygen species, and its subcomponent is what they tracked with one of the fluorescent antibodies. The subcomponent, I believe, was called GP91-FOX. And this is where they were able to show, hey, these mice, when you give them the virus with spike protein, they also have this inflammation in their lungs when they have the macrophages there, and they have, which they tracked with, I believe, green fluorescence, and they have that enzyme that produces reactive oxygen species, which they tracked with like a purple fluorescence, I believe. and this clearly indicates that the spike protein also induces high level of inflammation in its own right as well. Now let's go back to the mice that were mutant, which did not have any fibrinogen, so they cannot form clots. And these mice, if you put spike protein virus inside those mice, they don't have any of these problems. So they were able to show that such mice were not producing that inflammation. These macrophages were not coming to the site of injury, and they were not producing they were not activating this enzyme that was producing this reactive oxygen species. So really, really interesting. So clearly this somewhat triggered the authors into thinking a little bit more about this. And what they were able to show, and I thought this was fascinating, they wanted to see where does the spike protein actually even bind the fibrin. So what they did is they created something like 400 tiny small fragments of fibrin. So literally imagine they like chopping up the entire protein into 400 smaller fragments, except they actually had to make all of these, I believe. And they tested one fragment at a time. Does the spike protein interact with any of these fragments? So a lot of work, it's just an incredible amount of work. And they were able to show that the spike protein binds at 3 different locations on fibrin. 2 of these locations are at the D region. Remember in a previous video, I was telling you that D regions are involved in the formation of the D dimer. So 2 spike protein binds in the D region, and another binding site was somewhere between the D region and the central nodule of fibrin. Please check out past video to get a background on this. And that part is also known to already be involved in also cutting by plasmin. So remember, plasmin degrades fibrin, and this is how you remove clots. So clearly spike protein obscures potential cutting of clots by plasmin, and the authors indeed show that this is exactly what happens when you take clots and you put spike protein to it. You slow down the degradation of clots. And then those 2 fragments in the D region, one of them right away we'll see actually turns out to be very important. Why is that? It's already known that when you process fibrinogen into fibrin, and then fibrin is used for the formation of clots, you expose this smaller fragment that is highly inflammatory and is also potentially involved in causation of some of the autoimmune diseases, and manipulation of that fragment, whether you manipulate it genetically or you target it with something, you can prevent its negative outcomes. And that's the very same fragment that the spike protein happens to be interacting with. So they knew that some fragment, that fragment is interacting with the spike protein, but they wanted to find out exactly where does the spike protein interact with. So what they did is, this is really, really cool, really ingenious. They took that fragment and they remade it, except that they mutated 1 amino acid at a time along the length of that entire fragment. So remember, amino acids are the building blocks of proteins. You have about 20 of those, and that's basically how you produce proteins. Peptide is a small fragment of a protein. So they were using, I can't remember how big these peptides were, maybe somewhere like around 20 amino acids, 25 amino acids or so. And they mutated 1 amino acids from the actual sequence of fibrin, 1 at a time, to see if I mutate at this point, do I still see binding with spike protein or not? And they were able to show that some mutations along the fragment did not affect the spike protein binding at all. Some of them actually improved the binding, and then there was a region that when they mutated it, it prevented spike protein interaction, and they were able to find the exact location along that fragment where the spike protein interacts with it. And this was about 9 amino acids long, and this is actually really, really valuable. The reason why, because within that segment of 9 amino acids, that's the stretch that is known to be involved in formation of those autoimmune issues as well as those inflammatory issues. So what then the authors did, this is so incredible, they mutated those few amino acids that are known to be for the spike protein to bind that fibrin molecule. They mutated that in mice. So what does that mean? they basically mutated so that those amino acids were no longer there. They replaced them with something completely different in the genetic code of mice, so that mice were born that were mutant and no longer had that fragment working normally. It was completely like gone, so it could no longer be able to interact with the spike protein. And when such mutant mice that no longer had that fragment, normal fragment, were exposed to that pseudo virus, HIV virus that had the spike protein, once again they were able to show that such mice no longer responded in this abnormal inflammatory pathway. So the macrophages were no longer coming to the site of injury, and the reactive oxygen species was no longer also produced by that oxidase enzyme I was telling you earlier. So clearly what it means, what these authors were showing, is not only that the spike protein can produce abnormal clots, but also how it produces those clots, by even showing us exactly where the spike protein binds to the fibrin, and that if you were not to have this interaction, you can prevent all of this. Why is that important? Because the very last thing that they were able to show is, because this information have been known, that this component of fibrin can be so destructive, if you will, cause those autoimmune conditions and inflammatory conditions. An antibody was previously created that binds that fragment of fibrin and prevents all of that. I believe that antibody was called something like 5B8 or 58B. It's just like a number name. And they use that antibody. When they showed that when you take normal mice and you expose them to that antibody, you no longer had that problem at all in terms of spike protein inducing that damage in the lungs at all. So it actually works potentially as a therapeutic. This is the important part. And they were also able to show that in cells. So when you took simply in vitro, meaning in a like say a petri dish, you take your macrophages, immune cells, and you expose them to both the spike protein and that antibody, they were able to reduce that reactive oxygen species being formed. So clearly, this is probably the most valuable aspect, is because while we're seeing that we're having increased level of potential problems with the spike protein, they were able to show that there already is an antibody in existence that could act as a potential therapeutic to prevent all of this, or at least aspects of this. So I thought that was super important. What is weird about all of this is that this is not a new information at all. This paper I'm telling you about has been around for at least a year and a half. And the reason why we know about this is because it's a preprint, meaning it's still has not been peer reviewed. And why it hasn't been peer reviewed yet up to this point for so long, it's completely perplexing to me. I have no clue why such important work and such high quality work would not yet been peer reviewed and published in a peer reviewed journal. But thank goodness we have these preprints available to us so that we can actually learn about this valuable work that some of these authors are doing, and get access to it. That's all I have for you in this important video. And just, hey, listen, if you like this content, please subscribe to the channel, please share. This is how we grow. Leave a comment. We try to answer not all of them, because that's impossible, but a lot of them. And join my Patreon account. This is where I post even more controversial content. And also the other alternative is, please come to one of our COVID Q&A sessions. And if you want a free ticket to that, please subscribe to our newsletter. And the link for the newsletter subscription is in the description below. And we will be wrapping up this series soon. There's still a few more videos. This is how much content I was able to study over the last few months. And I think I'm gonna make one more video related to clots and spike protein, because there's just been so much science on this topic. And I look forward to seeing you in another installment. Bye everyone. Ciao for now.